you can consider this is a new beginning point, new turning point for your paper discussion. From this paper, I will be added some guessings. Actually, I cannot exactly say my structured essay questions are definitely will your structured essay questions in your exam paper. My structured essay questions will your essay questions, okay, in your A-level exam. Anyway, from this paper, especially do on a deep actually deep study on the especially on these essay parts. Hari ඔයගොල්ලන් සිංහල කතේරන කට්ටිය ඉන්නේ ගොඩක්. ඔයගොල්ලන් මෙතනදී මේ විශේෂයෙන්ම මට කියන්න බෑ structured essay questions තමයි ඔයාලගේ paper එකේ අනිවාර්යෙන්ම structured essay questions වෙන්නේ. මගේ essay questions තමයි ඔයාලගේ paper එකේ අනිවාර්යෙන් essay questions වෙන්නේ කියලා. හැබැයි ලොකු possibility එකක් තියෙනවා after doing so many analyses. يعني past papers ගොඩාක් මම analyze කර බලලා එන්න පුළුවන් විදිය possible වෙන questions තමයි මෙතනට මම දාලා තියෙන්නේ මේ මෙතන ඉඳලා යන paper series එකට structured essay questions ගත්තොත් අනිවාර්යෙන්ම lesson number 3 කියලා කියන්නේ structured essay වලටම create වෙච්ච lesson එකක් especially biochemical evolution part එක හොඳට බලන්න මොකද ඒක ඇවිල්ලා newly added එකක් syllabus එකට newly added එකක්. එතකොට lesson 2 වල goals, tumors, genetic and cancer cells also such regions, newly added regions, there is a huge possibility to answer. Okay, ගොඩාක් possible වෙන්න පුළුවන් අර අලුතින් add වෙලා තියෙන parts again, goals, tumors, cancers වගේ lesson 2 වල තියෙනවා වගේම lesson 3 වලත් අර ඒ වගේ තැන්. එතකොට මෙතනදී මම ඔයාලට විශේෂයෙන්ම highlight කරලා කියන්නේ මෙතන දැන් මෙතන ඉඳලා essay part එකට යන මේ questions තියෙනවානේ. ඒවට මම ඔයාලට දෙන marking scheme එකම ඔයාලා නිතරම follow කරන්න ඔයාලගේ note එක විදිහට. then ඔයාලට essay part එකත් විතරයි complete වෙන structured essay එකකට answer කරන්නත් පුළුවන් වෙනවා MCQ වලටත් ඒක helpful වෙනවා. හරි එතකොට මම ටක් ගාලා කියවගෙන යන්නම්. So these answers are available in your resource book. මෙතනදී ඔයාලා සාධක දෙකයි බලන්න ඕනේ. එකක් resource book එක අනිත් එක practical guide එක. ඔය දෙකෙන් පිට අහන්න නෑනේ. හරි එතකොට මෙතනදී මම හැම lesson එකක්ම cover වෙන්න දාලා තියෙනවා. අපි බලමු මම ටක් ගාලා answers ටික කියවගෙන යන්නම්. ඔයාලා බලන්න ලියලා තියෙනවද කියලා. ඔයාලා week වගේ දැනන lessons දැම්මම කොහොම හරි ඒ week ඒ lessons දැම්මම complete කරගන්න ඔයාලගේ exam paper එක looks like this. ඒතර molecular part එක අතරින්න බෑ, genetics අතරින්න බෑ, environmental biology අතරින්න බෑ, applied biology අතරින්න බෑ. ඒවා අමාරුයි කියලා. unit 5 කියලා කියන්නේ I think 50% of marks. lesson 5 වලින් lesson 5 එකක්වත් අපිට අතාරින්න බෑ. So we will start the discussion. Question number 1A again, part 1 and A. What is the sustainable food production? These 2 points should be included. The production of sufficient amount of foods for the human population. First one, the production of sufficient amounts of foods for the human population. This is the first point. And second point is by using environmentally safe methods. By using environmentally safe methods, the production of sufficient amount of foods for the human population using environmentally safe methods. Mention 3 steps based on the biological knowledge that can be used for sustainable food production. First one is production of high yielding varieties. Production of high yielding varieties of plants and animals. Production of high yielding varieties of plants and animals. Second one, production of disease-resistant plants and animal varieties. Production of disease-resistant plants and animal varieties. Third one, improve the post-harvest technology methods. Post-harvest means after harvesting. After harvesting, it's look like arranging or transporting like this. After harvesting the yield. Improve the post-harvest technology methods. Name a type of tetrose you know. That means 4 carbon. 4 carbon carbohydrates. Within the carbohydrates, especially in monosaccharides, we are talking about so many compounds regarding their number of carbon atoms. If 3 carbons, we call it as triose. The best example is glyceraldehyde. C4 we call it as tetrose. Only one example is erythrose. Okay, it is rare in nature. And C5 means pentose sugar. It's look like ribose, ribulose, like this. And C6 are hexoses, glucose, galactose, and fructose. What are the original compounds and what are the derivatives? Example, phosphoglyceraldehyde is a derivative of glyceraldehyde. Here in pentose sugar, RUBP, that means ribulose bisphosphate, is a derivative of ribulose, like that. So, answer is erythrose. Mention 3 works of cilia and flagella. In both cilia and flagella, same arrangement. Same arrangement is there, but there is a difference. If this is a bacterial cell, if this is the cell wall, the next membrane we call it as plasma membrane. Cilia, both cilia and flagella started from the plasma membrane. This is the flagella and this is the cilia. Cilia are high in number, but the length is higher than cilia. Length of the flagella is higher than cilia. Okay, so commonly what are the functions? First one, act as a locomotor appendages. Act as locomotor appendages. Second one, can move fluid over the surface of the tissue. Can move fluid over the surface of the tissue. Third one, cilia lining in oviducts. Cilia lining in oviducts. If this is the oviduct, this is the ovary and ova is produced. This is the cilia lining in the oviduct. This is the oviduct now. So it helps to move the ovum into the uterus. So cilia lining in oviducts help to move egg towards the uterus. So what are the 3 functions? Act as locomotor appendages, can move fluid over the surface of the tissue. Cilia lining in oviducts help move an egg towards the uterus. What are the components of the plasma membrane? You can say mainly phospholipid bilayer, or you can say lipids and proteins. Mainly it is composed of lipids and proteins, especially phospholipids. What are the peripheral proteins of the plasma membrane? So, if this is the phospholipid bilayer, of the plasma membrane, some proteins are fully embedded, okay, within this phospholipid bilayer. But peripheral proteins looks like this, okay? Peripheral proteins look like this. So you have to write proteins which are not embedded in the lipid bilayer. Proteins which are not embedded in the lipid bilayer and are loosely bound to the inner surface of the membrane. If this is the extracellular space, this is the intracellular space. That means this is the inner surface now. So this is the inner surface of the bilayer. So you have to write proteins which are not embedded in the lipid bilayer and are loosely bound to the inner surface, of the membrane called peripheral proteins. Those are the in your answer, these tissues. Parenchyma tissue, collenchyma tissue, sclerenchyma tissue, xylem tissue, phloem tissue, and epidermis. Those are the answers. Mention 3 types of tissue containing systems you above mentioned. So initially you have to write the ground tissue system. These 3 are included in the ground tissue system. Then vascular tissue system. Then the dermal tissue system. So what are the 3 tissue systems? Ground tissue system, dermal tissue system, and vascular tissue system. Which reagent is used to identify starch in the science lab? Iodine solution. What could be the observation of above experiment? Dark blue, dark blue. Which 2 solutions should be used in addition to the laboratory experiment in which the activity of an enzyme is studied using the test in 1? Amylase solution and starch solution. Improve the experiment you mentioned in 2B and explain what your observation at temperatures of 5 degrees Celsius, 27 degrees Celsius, 40 degrees Celsius, and 60 degrees Celsius in the experiment that studies the activity of an enzyme with temperature. So can you remember practical of studying the enzymatic activity and as its modification factors, especially the temperature, how the temperature is affected for the enzymatic reaction. For this one, the memory of iodine identification also used. You know, starch plus iodine means starch iodine complex, dark blue in color. Okay, dark blue in color. And starch is broken into the maltose by amylase enzyme. Okay, you know amylase is an enzyme. What would be the temperature in degrees of Celsius and the rate, enzymatic rate? The graph should like this now. This is the critical. This is the critical temperature. It is about 40 degrees Celsius. Now, so in here, initially in practical, we have to prepare 2 different solutions of starch solution and amylase solution. Okay, then we will mix them together. That means if this is number 1, if this is 2, 2 is added into 1. Okay, in this time the stopwatch is started. After adding 2 into 1 means what happened? Starch becomes maltose. Okay, with the time. Initially, we take a porcelain tile, white color porcelain tile. Why we use a white color porcelain tile? For better observation. Okay, better observation of color change. So initially, we take a drop of this mixture into this tile and we add iodine solution. Then we can observe dark blue coloration because initially starch is a little amount of starch is converted into maltose or no any starch is converted into maltose. But with the time after 5 minutes, okay, after 5 minutes, again we take a 1 drop of this solution and put into this porcelain tile and add I2. Then we can observe the intensity, the color intensity of blue is reduced. Then what happened? Amylase amylase do its function. Okay, amylase do its function. That means in here, with the time, most of the starch solutions are broken into the maltose. The experiment, starch and iodine is identical to each other. That means iodine cannot produce dark blue coloration with maltose. Okay, only with the starch. So with the time, starch molecules are reduced into maltose. Therefore, the dark blue coloration, intensity of dark blue coloration is reduced. So after about 15 minutes, when we take one drop of this mixture and put iodine into this, no any color change, we can observe the color of iodine, that means brownish yellow, yellowish brown, no? Yellowish brown color from this drop, because all the starch molecules are broken into the maltose. Therefore, no reaction with iodine and maltose, no any color change with maltose and iodine. So we can observe the enzymatic activity. As a modification, we change the temperature of this mixture for different experiments. So what would be the observation? Initially we have to prepare a starch solution and amylase solution. I think if starch is I cannot remember exactly. Please refer your practical guide. I think starch is about 10 milliliters, amylase 5 milliliters. I think according to my memory. So as the modification, we prepare a water bath in 5 degrees Celsius. Okay, and take two samples of starch and amylase and mix together and keep it in 5 degrees Celsius condition. Then we do the same practical, okay, for so many times with so many temperatures. That means after 5 minutes, we take one drop of this mixture and add I2. Okay, then we remove this one, and again we add 10 milliliters of starch and 5 milliliters of amylase, then again prepare a mixture. Then the temperature should 27 degrees, that means room temperature. Here again we add I2, and as the next step, the water bath should be heated, then keep 40 degrees Celsius, and again newly prepared mixture is kept in your 4 degrees Celsius water bath. Then we take one drop of iodine and I2. So here we can see light intensity, blue color intensity is decreased because here same time intervals after 5 minutes, 5 minutes, 5 minutes. No effect on the time duration. So why the light intensity is decreased? Color intensity is decreased because this we can explain by using this gap. Okay, to the 40 degrees Celsius, when the temperature increases, the enzymatic reaction is increased. So after increasing enzymatic reaction, starch is converted into maltose rapidly. Therefore, here color intensity is decreased. So once the 40 degrees Celsius is passed, what happened? Proteins are denaturated. You know, proteins are made up of enzymes. Then enzymes are denaturated. Then proteins are denaturated. even if you have increased the water bath temperature as 50 degrees Celsius and again prepare a new mixture by using starch and amylase and add one drop into the porcelain tile and add I2, then we can observe um, dark blue coloration. Again, dark blue coloration, or again the intensity is increased because enzymes are denaturated, they are not worked. So how we can explain it? Answer should, when the temperature is increased, from 5 degrees to 27 degrees to 40 degrees, the decreasement rate of blue color intensity is increased. The decreasement rate of blue color intensity is increased. After passing 40 degrees Celsius, when the temperature increases, the blue color intensity is increased. So that is the explanation. When the temperature is increased from 5 degrees to 27 degrees Celsius and then to 40 degrees Celsius, the decreasement rate of the blue color intensity is increased. After passing 40 degrees Celsius, when the temperature increases, the blue color intensity is increased. Okay. Mention the functions of epidermis of plants. Answers are available in your resource book. So how we can write? Defense against physical damage and pathogens. Okay. Cuticle helps to prevent water loss. Root hairs involved in absorption of water and mineral ions. Guard cells help gaseous exchange. Hair-like trichomes reduce water loss. Shiny hairs reflect excess light. Some trichomes secrete chemicals involved in defense against insects, pathogens, and herbivores. What are the main parts of basic structural unit of nervous system? It is neuron. So what are the three parts? Cell body, dendrites, and axon. Cell body, dendrites, and axon. Cell body, those are the dendrites, and this is the axon bulbs. Okay, so main regions is cell body, dendrites, and axon. Mention all the function of glial cells by mentioning other name. Other name is neuroglia cells. And what are the four functions? Nourishment of nerve cells, insulation of nerve cells, insulation of nerve cells, replenishing neurons, sometimes modulate neuron functions. Those are the four functions. State the differences between simple columnar epithelial and pseudostratified columnar. State the different position. Okay,